All right, so now let's run through a quick comparison of mitosis versus meiosis. So, starting with synapses, all right, we're going to basically go through the different processes that occur in each one of these, but compare them going from one to the other. Sound good? So, in mitosis, first of all, does synapsis occur? And if it does, when does it occur? All right, so looking across the board, in mitosis, does synapsis occur? No, because during mitosis, we do not have that homologous pairing of chromosomes. Remember, the purpose of mitosis is to produce perfectly duplicated cells. So we don't want to risk any crossing over events happening, right? Moving on to meiosis 1 though, does synapsis occur? Yes. And when does it occur? Right, prophase 1. And then in meiosis 2, does synapsis occur? No, right? Same thing as in mitosis, all right? The only difference really between mitosis and meiosis 2 is that in meiosis 2 we are dealing with haploid cells, whereas in mitosis we're dealing with diploid cells. But otherwise, the processes are basically the same. And in meiosis 2, there really actually isn't even an option for homologous chromosomes to pair up. Moving on to crossing over, right? We kind of already touched on this, but does crossing over occur in mitosis? No, it does not, right? We want those cells to be genetically identical to one another, and crossing over increases genetic diversity, so we don't want that to happen in mitosis. However, in meiosis 1, yes, we do see this occurring, and it happens during the same phase that we see synapsis, right? So homologous pairs of chromosomes synapse together, and once they are linked together via synapses, they cross over parts of their chromosomes and exchange genetic information, right? We also call that recombination. And in meiosis 2, once again, you might start seeing a pattern. No crossing over occurs. All right, now what separates during anaphase? This is something that a lot of students struggle with. So during anaphase of mitosis, we have sister chromatids are being pulled apart, right? And contrast though, in meiosis 1, since we have that homologous pairing, that synapses occurred in prophase 1, what gets pulled apart during anaphase 1 is homologous chromosomes. All right, so full chromosomes are separated from other chromosomes during meiosis 1, whereas in meiosis 2, we're back to splitting those sister chromatids. And then looking at independent assortment, does it occur and when? In mitosis, we do not see independent assortment because independent assortment is defined as the separation of homologous pairs being random along that metaphase plate, right? So the homologous pairs of chromosomes line up in a random order. On one side, you'll have either the maternally inherited chromosome or the paternally inherited chromosome that are paired together, and the side on which they align is completely random. Therefore, which side of the cell they end up getting pulled to is also completely random. So since there's no homologous pairs that we're dealing with in mitosis, we don't see that. However, in meiosis 1, we do see that, and it's during metaphase 1. In meiosis 2, you got it. We do not see independent assortment. Now, are the cells that are produced from each of these phases genetically identical? In mitosis, yes, they are genetically identical. However, in meiosis 1, the end of meiosis 1, we do not have genetically identical cells, right? Because we had crossing over and independent assortment to aid in that genetic diversity. And at the end of meiosis 1, our cells are no longer diploid, right? So that is another difference between our current daughter cells and the original parent cell. In meiosis 2, we do not have genetically identical cells either, right? Same things. Back in meiosis 1, we had that independent assortment and recombination, and because of that, once we get to the end of meiosis 2, we no longer have genetically identical cells, and our cells are haploid, right? So speaking of ploidy, let's go through it. In mitosis, we start with a diploid cell and we end with a diploid cell. Perfect clones. Whereas in meiosis 1 and meiosis 2, a little bit different. In meiosis 1, we go from a diploid cell to a haploid cell. And in meiosis 2, since we're starting with a haploid cell and we end with a haploid cell, it's haploid to haploid. Sound good? Okay, so I'm now going to bring up a couple of key questions that students sometimes struggle with. So let's go through those together. Is the cell that you see on the screen right now in the middle of mitosis or meiosis? And how would you know? Feel free to pause and give this a go. Right, this cell is in the middle of meiosis. We can tell because we can see that there are pairs of chromosomes along that metaphase plate, right? We have one orange chromosome, one red chromosome, and they're paired together. So that tells us that we are in meiosis because mitosis does not deal with chromosomes in pairs like this. And now for this one, what stage is this cell in and how do you know? Right, so we know it's in meiosis, but which stage of meiosis? Well, we can see that chromosomes are lined up along that metaphase plate right down the middle of the cell. So now we have to narrow it down between either metaphase 1 or metaphase 2. Well, let's think about it, right? Which metaphase do we still have pairs of chromosomes? Right, during meiosis 1, so that would be metaphase 1. And now here's something that sometimes students get confused on. Does crossing over occur during this phase? And the answer is no, right? Meiosis has crossing over, but only during prophase 1 of meiosis. So how would this image look differently if it were in metaphase 2 of meiosis? Basically, we would see single chromosomes as opposed to homologous pairs of chromosomes, right? So here we have our homologous pairs still. They're still together, so we know that this must be metaphase 1. But after meiosis 1 ends, we've already separated one of the pair to one side of the cell and the other part of the pair to the other side. So they are now in two separate cells, right? Here's our dark purple friend, and here's our light purple friend by the time we're in metaphase 2. So in metaphase 2, we just have single chromosomes lining up on that metaphase plate, very similar to what we see in mitosis. So how would this look differently if we were looking at metaphase of mitosis? Basically what we just went over, right? Instead of seeing these pairs of chromosomes along that metaphase plate, we would see single chromosomes instead of pairs, right? And so it would look something like this if it were a mitosis, right? If we want to go back, we can see that we had previously the pairs of chromosomes, whereas here we have single chromosomes all lined up along that metaphase plate. Very similar to what we would see in meiosis 2, except for in meiosis 2, the cell is haploid instead of diploid. All right, so now we're going to break off into a little game. This next part is totally optional, but I do highly recommend it if you want to see real life examples of a cell going through mitosis or meiosis, right? The examples that you've mainly been seeing so far are of graphics, right? They're basically of diagrams of cells, whereas these will be real life cells under a microscope. So this is a really good opportunity for you to practice if you have the time to do so.